air. And let me just verify that stream's in good shape. Dashboard, no data.
You realize this thing is slowly taking over our lives now, you see. It was originally like, I'm just going to stream, you guys won't even know anything about it. Now it's like, we're all beholden to the stream. Slow.
Oh boy, now I need to beholden to the stream.
Wow, it sounds like there's 50 of us in the room.
I don't know why I'm getting that audio, but we are live.
All right, should we go?
Yes.
All right, we'll go.
Please go.
So this is just very informal. This is a continuation of what I was asked to do, to do some neuroscience talks, some basics. So we talked about the neuron before. We've talked about columns and mini columns. I was asked to talk about pooling, especially temporal pooling, and also something about hierarchy. So I thought I'd combine those two today. But anything you want to ask about, just ask and we can divert. It's not like a highly structured thing. So most of these pictures on the board are about the anatomy of hierarchy. I'm going to talk about pooling just sort of ad hoc here in front of you. I don't know how most people use the word pooling. Right? It may have a definition in machine learning that I'm not aware of. So I'm just going to talk about how I think about the word pooling and what it means to me and where the thoughts about it come from. And it may be consistent with other people thinking, it may not. I just don't really know if the term is used elsewhere very heavily, or if everyone uses it the same way. So to me, pooling is a very simple idea. It's a many-to-one mapping. It's like many patterns map to fewer patterns. And that the brain has to be doing pooling. You can simply think about it this way. What if I give a million images and I'm classifying them into 10 categories? And I'm doing this in some sort of neural network. And so I can put in a million different patterns and I'm going to get 1 of 10 outputs of this thing. So that is a pooling operation. In some sense, in the brain, I would have, at some level in the brain, I'd have neurons that represent the classification of that image. And that would be a pattern. And there's many millions or hundreds of thousands of patterns which map into that. And so along the way, you have to have changing patterns and a stable pattern, changing patterns and a stable pattern. And that's the concept of pooling. It's a many-to-one mapping. I don't know if that's consistent with everyone else's understanding of pooling, that most people use it. So clearly it's happening in the brain big time. Even something like when I just look at a scene or look at an image, your eyes are moving constantly, yet the your perception of the image is stable. And that also requires some sort of pooling because there's some stable neurons which are representing what I'm looking at, and the inputs are changing underneath it, so there's a many-to-one mapping. And this many-to-one mapping is not a random thing, it's a very precise thing. It's not like I'm just mapping willy-nilly to something. These are very precise patterns and many precise patterns. So when you think about pooling, there's two basic ways you can think about pooling, and the terms we use for that are spatial pooling and temporal pooling. And they're pretty simple in concept. Spatial pooling is the idea that, let's say I have some sparse pattern, some SDR here, and I'm mapping it into another pattern. So this is my many-to-one mapping. And so this is sparse, so it's going to be some sparse set of ones and lots of zeros, something like that. And I'm going to map that to another sparse pattern. If I just change one of these bits, I generally don't expect this output to change. So you might think of that as noise, you might think of it as a slight variation, but if I have 1,000 bits or 1,000 ones, if I change some of them, I don't expect this output to change every time I change one of these bits. And so that is a form of pooling. It's essentially saying, hey, maybe I'm mapping this pattern to that pattern, but any small variation, if you think of this pattern as a point in a high-dimensional space, if I'm nearby that point, that all my ones haven't changed. Only a small subset of my ones have changed. So if I have 1,000 ones, maybe 100, up to 100 can change. I'm just moving locally in that point in high-dimensional space. I still get the same output. So that's why the term spatial pooling came about. Because you're spatially close to the original at some point. These a bunch of points that are spatially near each other in this high-dimensional space are all going to map to the same point. It doesn't require any learning to do that. You can do this with a random you have neurons up here that randomly get inputs from some subset down here, and as long as, and then you enforce a sparsity function on this. There's a competition going on between who gets the win, who gets the most inputs. And if you do this randomly, you'll get spatial pooling. In fact, we did a lot of early experiments where the spatial pooling didn't learn at all, and they worked great. So it's just a natural consequence of sort of distributed neurons with needing multiple synapses to fire. We did a lot of work on training the spatial pooler, and you can get better results when you do that, meaning these synapses can be learned, and so I won't go into that. But that's basically, spatial pooling is essentially just a very simple mapping using neurons from one pattern to another pattern, and naturally there's a lot of patterns that are close to this original one that will map onto the same one, so that's a form of pooling. All right. The second type of pooling, the temporal pooling, is far more interesting and difficult. And we use the word temporal because we were thinking about it in the following way, that you might have, in this case, you're going to do, let's say, here's SDR1 and SDR2 and SDR3, and these are completely orthogonal as much as any SDRs are orthogonal, right? So they're orthogonal vectors, and not completely, but they don't overlap much. And they're all going to map to the same one up here, right? And so one way that the brain does this is say, well, because they're a temporal proximity, this one follows that one, this one follows that one, we can assume that it's likely that there's a common cause in the world, and so these three different patterns that follow each other in time actually do all represent the same thing. So you can think like notes in a melody, right? Well, they're right after one another, so they probably represent the same thing. And so the same thing would be, the basic same thing would be true if I'm trying to touch a coffee cup or move my eyes over some object. You're basically assuming that there's a temporal correlation corresponds to a common cause, and that's where the word temporal pooling comes from. This, of course, has to be learned, that these neurons up here that have there's some pattern that's stable up here, and you got a different pattern here and a pattern here and a pattern there. These neurons up here have to learn to recognize that pattern, they have to recognize that pattern, they have to recognize that pattern. And so you can present any one of these patterns and you'll get the same one up here. So it's a many-to-one mapping. These are, of course, the same neurons here, meaning this is a subset of the active neurons, the same neurons, a different subset, the same neurons, a different subset. So it's still one population to another population, but we're going through different patterns down here, and they have to learn them up here. So that's the concept of temporal pooling. They are bound together because of temporal proximity. Does that make sense? The next thing is we can break that into two different parts. High-order sequences and sensory motor sequences. So basically these could be like notes in a melody, and that's all you need to know. Or they could be you're getting different changes, your inputs are changing because you're moving. So in one case, the world is changing, in the other case, you're changing. And this is when we focus on this one first, and this is where we came up with the temporal memory algorithm. HTM, temporal memory algorithm. And I'm not going to walk through that now, but how you use the same set of neurons to represent a sequence uniquely, so that the third note in some melody is represented uniquely, so that you could say, I could classify that all on its own. I know that's the third note in this melody, and that is going to be unique to that melody, and therefore I can classify like that. And this was, of course, when you're moving, so this is more of the sensory motor. This is what we did with grid cells and our more recent work. So I'm not going to go through those mechanisms here. I'm just pointing out that those are two different types of temporal patterns, and you still have this many-to-one mapping. Okay, so that was my introduction to pooling, and before I go on to talk about where it's happening. So any questions about that?
So does spatial pooling and temporal pooling happen like between the same populations? Because it's both.
Okay, good question.
Okay, yeah.
Let's get to that.
Okay.
Okay. Is there more to your question?
No, that was basically. I mean, you get it already, and yeah, I thought we were going to elaborate.
Well, I don't. Let me just jump into the next stage here, and we can talk about what we think is going on. Okay. Where it's occurring. Is it the same set of cells or not the same set of cells? That was your question?
Yeah, basically, yeah.
Yeah. So, all right, so now, Simtai, if you want to step out, you can, but
That's fine.
he wants to get other work done. He doesn't have to. So now I'm going to talk about architecture of the cortex and where this might be occurring in the cortex and hierarchy in the cortex, okay? And where the neurons for this are. So
maybe one quick clarification that might be helpful is, because it's been confusing in the past, the process of doing the high order sequences or the sensory motor sequences is completely independent of the pooling,
yes.
Pooling operation.
Oh, that's right. Yeah.
In the white paper, we use the same term for both, which was confusing.
That was back in the white paper.
Yeah.
We ruled that day. We wrote that, right? So you're right. This
it may have gotten a little confusing.
So how would you clarify that? What do you want to say? Go ahead and say what your clarification is exactly. I don't know if that was captured on the stream.
Sort of, if you're doing high order sequences here, this block here is making predictions of the next input that are coming in, and it's creating representations that are specific to that point in the sequence. So that's all going on here. And same thing with the sensory motor here, making predictions about what you're going to see, and it's constantly changing. And both of those would be input to the temporal pooling. So that the stability.
Temporal pooling applies.
It's not like these are subsets of temporal pooling.
No, these are two different. Right, I'm sorry. So these are two different ways you can generate the patterns that have to be pooled.
Yeah.
That's your point. Yes.
What is the
difference? It's just
the top one is a stable pattern that represents. Let's say you have notes in a melody, A, B, C, D, E, F. And each of those have points in the sequence, but the song would be a stable pattern.
This is the name of the song,
okay.
and this is the sequence. This is the notes that are coming in. But the way the temporal memory system works is as you get the notes, we represent them using the mini column thing, where they're unique representations. You remember this? It's part of the quiz. Now you're showing them. Now you're going to regret not getting that your thing. That we can go through that as a separate exercise. The point, the important point here is you've got these changing inputs coming into the brain, and they could be because the world is changing, like you're hearing the notes in a melody or a bird flies by, or they could be changing because you're moving, and that when you move, the inputs change. So I'm moving my fingers over the coffee cup. I'm moving my eyes. In both cases, you have a changing input. In both cases, we have to come up with representations for that input that are unique to the specific object. So in the temporal memory, we end up with these very unique SDRs that represent the note, like it's an interval in the song, there's a third interval, but it's on this particular melody at this particular point. So it's unique. And the same thing here, when I touch the coffee cup, I just don't want to have the input. I need to make it unique to this object, and so that's the whole grid cell hypothesis. So that, and I'll come back to that in a moment here, that you have to convert these into very unique sparse patterns, and then you can pull them together to represent either the melody or the coffee cup or something else. If that's
a very primitive form of that happens in CNNs too. Like,
I was wondering about that. Where does it happen in CNNs?
They call a max pooling operation, which so you have, let's say, four things would feed into a max pooling, and it would take the maximum of the four inputs coming in. It's like a very primitive.
But how's that pooling? It's just taking the maximum.
It's like a K winner take all with K equals 1 and 4 inputs.
But do all four of them map to that same pattern?
Yeah, so it'll just take the maximum value and pass it on. So but in a CNN, imagine you shift the pixels by 1, shift the image by 1. The output of the max pooling will be more stable than the output of the input. Right? Because the input, everything will change in the input when you shift by 1 of the CNN output. And then that goes through a max pooling, and because of the max pooling step, that'll be a little more stable.
Maybe someday you could explain that to me in more detail, not right now, because I'd like to understand that. To me, this was a guiding, fundamental observation about brains that in my mind goes back over 30 years. Like the brain has to figure out how to take all these changing patterns and bring into unique stable ones.
Yeah.
And that's sort of a key principle, and it has to do a lot of this. It's not like a fuzzy operation. It has to be a very precise operation, and it's going on everywhere. And
so it'd be like Hubel and Weasel, simple cells, complex cells. The complex cells might be a little more stable as the inputs are.
Yeah, but I think this is much more fundamental than that, right?
Yeah.
This is like literally completely unique patterns that are not related. They don't seem to be related at all, but have to be pulled together. Right. They're not overlapping at all. So to me, these things have to occur. It has to. If CNNs are able to classify images, they have to be doing pooling by definition. But they don't have an explicit pooling step. But maybe the max
pooling is a
kind of like
is the closest.
You have to think about it. But in the neurons, it's a very precise. Like many patterns map exactly to one pattern, and it's a learned operation. That's what temporal pooling does. Okay, so this has to be occurring in the brain everywhere. I mean, if we're to recognize melodies, to label anything, to create stable representations of anything, then to know that these changing inputs on my finger are a coffee cup, this all requires pooling and temporal pooling. So now we're going to turn our attention to the cortex. Okay, so we all know that the cortex is a big sheet, right? And it's divided into regions, but the regions all look very similar. Here's a picture of a sort of a characterization of a slice through the cortex. So this would be like 2.5 millimeters thick in a human. And these little, I've marked up 1, 2, and 3, those would be like regions of the neocortex. Of course, they'd be much bigger than this. So it's 2.5 millimeters thick. V1 might be like that big, or something like that in a human. But imagine if you're slicing it through it, and so these regions look very similar. But if you follow the information flow, like this is sort of a classic view going back to the 60s. You might say, oh, the information comes from the eye, and then you have 1 million fibers coming off the back of the eye. They make this relay stop here in the thalamus, these relay cells in the thalamus, and then they project to the first visual region of the cortex.
So that's V1.
That would be V1 in this case, if that was from the eye. And then V1 has a lot of fibers which exit the cortex, go into the white matter, and then project to V2. And then V2 has a lot of neurons that exit into the white matter and go into V3, and so on. In about 3 or 4 steps, you start having cells that seem to recognize big objects over larger.
And one is outside?
One is, in humans, it's at the very back of the brain and kind of folds into remember the brain kind of here's a moment.
Alright, so as it goes deeper, it goes inside the skull, right?
No. Oops. I gotta glue this one together.
No, don't glue it. I like it.
Yeah, well, it'll always fall apart. So this V1 is like back here, like right around here in a human, but it also goes into this fissure here.
Right.
And then there are different, where the other regions are, it doesn't really matter. It's just the connectivity of all that matters.
Oh, okay.
Right. It turns out that V2 will be adjacent to V1 and V4 will be adjacent to V2, but it's really messy.
Okay.
So the adjacency is not that essential.
Okay.
What's essential is the connectivity, and obviously you have this very complex map. You can't make everything adjacent to everything else.
Okay.
So then there's regions that project far away and things. And there's, of course, there's 2 different visual streams. There's a ventral stream and a dorsal stream.
So this is all the classic view.
This is the I'm going to talk about the classic view. Then I'm going to talk about the reality view. But just starting with classic, this is when people looked at this and they said, well, how is the cortex wired? This is the first thing they saw, and they saw a lot of these connections. These are big, they're very prominent. They said, that's how it looks.
And is that constant? Is that given? Or are there cases where the connectivity just looks very different?
No, this is pretty constant. I mean, every human looks the same pretty much. The size of these regions can change quite a bit.
This is the classic view. The real view is more complicated.
Yeah. But the basic region-to-region architecture doesn't change in your lifetime.
Okay.
It can, if you were born without eyes or without a visual input, then these regions can become something else. They become tactile regions or something like that. But if you're a normal human, everybody has these same regions, as far as we know, and they're all basically connected the same way. There's variation. One of my most fascinating facts I would mention is the fact that the size of V1 in a normal human can vary by a factor of 3. And so the size of these regions can be quite big or smaller, but and they have normal vision. So Anyway, so this is the classic view, okay? So, and unlike a CNN, you know, the classic view is you can get object recognition in maybe 3 or 4 steps. So you don't need 100. So that's always been a little bit of a mystery. Now, if you look at actually in this classic view, what does it look like? Where are these cells? And this is getting closer to your question, Michelangelo. So here I've labeled the 6 layers of the cortex. That's a misnomer. We still use that term, 6 layers, but really, there's lots of different ways you can decide the layers, and there's many more different types of layers than 6. So, but we still call them the 6 layers. But there's many different types of cells in here. But there is a laminar structure. And the classic view is the input that comes into a region, any one of these regions, this input here, this input here, this input here, terminates primarily in layer 4. That's your input layer. Layer 4 projects to layer 2, 3. So that's something's going on there. And then layer 2, 3 is one of the output layers of the cortex. So the cells in layer 2, 3 exit, go down, and go up and project to layer 4 in the next region. So this exists, but it's a subset of what's actually going on. So this makes it look pretty simple. You can say, look, every region it's got an input. There's basically 2 layers of cells involved. Then it goes up to input, 2 layers of cells, and so on. If this was your view of the world, then temporal pooling has to occur, all these functions have to occur in these 2 layers. And that's the way you look at the cortex. And so that's sort of like saying, okay, well, you're obviously saying, what about these other layers? We'll come back to it in a moment.
Maybe I point out an analogy to machine learning. This is the basic inspiration for CNNs, convolutional networks. The idea that you have input coming in. This is your quote-unquote simple cells recognizing features. Yeah. This is your more stable complex cells, where the max pooling might occur, and then you go on and just connect with layer 2.
Yeah, so that's great. And then what we did, perhaps maybe the convolutional neural networks didn't do this, we came up with a mechanism that said, all right, we could model in just a single layer this high order sequences. We can say if I just break out that one layer, then I can put these mini columns in there, and we learn high order sequences in that. And we'll go through that mechanism now. And what we didn't do for a long time is we actually didn't do the temporal pooling layer. You know, the temporal pooling operation is not necessary to learn the sequences. It's only necessary to create a stable representation of the sequences. But we have subsequently done that in some of our other work related to sensory motor inference. So we could create a temporal pooling layer here and a sequence memory layer down here. And what you would end up is you'd end up with a stable representation for a sequence. And then a new sequence comes in, you have a stable representation for the sequence. So this is the name of the melody and the object. And those 2 operations could occur, and then you could do it again. But it wasn't really clear why you do it again and what's and so on. There's a lot more to it. But anyway, that was the basic idea. So one could argue that perhaps layer 2, 3 is a temporal pooling layer. Now, again, I say layer 2, 3. Many early papers many current papers don't make a distinction between layers 2 and 3. Layer 1 actually has very few cells, so they sort of ignore it. It's more there are cells in layer 1, but they ignore it anyway. But some people can differentiate all kinds of cells in layer 2 and 3. So it's maybe not just one type of cell. There's like lots of cells in there. And so there's layer 3A and 3B, and layer 2A and 2B, depending on who you ask. But very often people say, well, it looks like there's a lot of connections to layer 2, 3. Those become the output to the next region, and so on. That was the classic Hewell and Basal, or back in the 60s, feed-forward pathway. There is also a classic, in this view, there's a feedback pathway. And the feedback pathway starts with cells in layer 6. And they project back through the white matter into the previous layer. Previous region, so I'm not showing that here, but it would be a backwards projection. And in the original classic view, what these axons do is they spread very long distances in layer 1. So it's a feedback, and the 2 cell types that connect to it would be the layer 5 and layer 3. These cells, these are parental cells which have these long apical dendrites that extend up into this layer 1, 2 area, and they, in theory, could make connections on their apical dendrites to this feedback. So, yeah, Lucas.
Is there feedback only between regions, not between layers?
Oh no, there's a lot going on between layers. I haven't gotten there yet. Tons and tons. Okay. Most of the connections in the cortex, most of the axons, most of the dendrites are all local. Tons of things going on there. I'm just talking about region-to-region connectivity at the moment.
Okay.
This is, when people talk about the hierarchy of the cortex, they're talking about region-to-region hierarchies. This is one region, the next one up the hierarchy, next one up the hierarchy type.
Okay. And the layers are, they're stacked, right?
They're stacked.
Okay.
So you can.
They're physically. Like
When you look at the hallway at the top of the stairs, that's a picture from Cajal in 1900, and that's a picture that Cajal saw through the microscope, and he said, "Look, there seems to be layers here." And why do they see it? Because the cells, some of the cells are bigger, some of them are smaller, some are densely packed, some are not densely packed. So the first thing they did, they just looked at these stains of the cortex. And they could say, looking at those stains, they look like these layers, these laminar layers. Nowadays, you can look at the connectivity, and the connectivity implies a different set of layers, or an additional set of layers, or they can look at gene expressions and so on. So there's many, many different ways you can determine what's a unique cell type. But in theory, you would say there's a population of cells that look similar, they project someplace similar, they can input someplace similar, they express similar types of chemicals, therefore that's a layer. But I haven't gotten to that yet. There's almost all the complexity in the brain is in here. And these connections represent a small percentage of all the connections that actually exist in the brain. But if we're talking about region to region, so in a convolutional neural network, your layers are very simple, and there's 100 of them. Here, this region, which is sort of equivalent to maybe a layer in a convolutional network, is very complex, and there's only a few of them.
That's my less than 1% number.
Yeah.
Talk about this. So this is the classic view of the hierarchy and how processing occurs, but it accounts for less than 1% of the synapses in the neocortex.
Yeah. Yeah.
But it does happen, right?
It does happen. This is here.
But it's the same way as you have a hierarchy in a fully connected graph. If there is a hierarchy, but if you ignore the fully connected part of it, it's kind of yes, it exists, but there's 100 times more connections than that.
So what you're saying is a lot more sparse between regions than it is within
No, that's not just about sparse. Remember, there may be,
synapses. There's a count of synapses, connections. Yeah.
I mean, there may be, in a human brain, there might be 100 regions. It's debatable.
Okay.
What's a unique region? It's very hard to tell sometimes. But let's say there's 100 regions, and there might be, any region might get input from 7 or 8 other regions, actually. It turns out to be more than they thought. And so the connectivity of the region-to-region graph is closer to 40%. But that doesn't tell you how many axons and synapses. It just says, if I just look at the connectivity of the graph between these 100 regions, it's maybe about 40%, which is much higher than people would have thought.
Right.
But the actual number of synapses is small. As soon as I was saying, mostly the activity is occurring in here. I mean, because remember, these connections, there might be a million fibers going from here to here, and a million fibers going from that kind of thing. But there's hundreds, there's trillions of synapses in here inside of this.
So from neuron to neurons, it's fair to say that between layers it's less sparse because we have more synapses than between regions from neuron to neuron.
I don't think we should say that. I think the only thing we should say is that, of course, activity is always sparse,
right,
everywhere we look, pretty much. And the connectivity sparsity is very difficult to put your finger on because some of these cells project long distances, some of them only project locally, so sparse compared to what? What population are you looking at? Right. I don't find it fruitful to think about that connectivity right now. Like I can say something concrete about the sparsity. In general, if you took any two neurons, even nearby, they're unlikely to connect or have very few connections between each other. Two neurons that are right next to each other in the cortex may have 1 or 2 synapses, between them out of 10,000 or 5,000. So the connectivity graph could be large, but the sparsity of connections could be small. I mean, who knows? Anyway, just trying to get to this sense of this region-to-region connectivity. This is this classic view. It looks pretty simple. You have 2 layers, project to 2 layers, project to 2 layers. There's a feedback thing. People say, hey, maybe that's doing, back prop. No, but maybe they think it is. So that is all true, but it's really, really not the whole picture, not even close. But it is instructive because these connections are large and they do exist and they were proven and they're still there. And one can say, well, there's something going on because I get an input to layer 4 projects to layer 3 projects to layer 4. That is there, so we have to explain that. Okay. There's another feed-forward pathway which was ignored for a while, or it wasn't understood. And two scientists we talk about here a lot, Murray Sherman and McGillivray, have been promoting this idea. And here's one way to think about it. See over here, there's the input from the eye goes to this thing called the thalamus before it goes to V1. Well, this is true for input from the ears and from the skin and everywhere else. In fact, every primary sensory region gets an input from the thalamus. So that's pretty important. It's obviously doing something important. What they pointed out was that's actually the wrong way to think about it. If you look at the thalamus has lots of these little subregions in them. And it turns out that if I follow a different way to look at this connectivity, an additional way to look at this connectivity, is that I get the input from a sensory organ, such as the eye. It goes to the thalamus. It then projects to some region. The output of this region, a different output, not the same one we had over here, a different one, leaves this region, goes to a different relay center in the thalamus, which projects to the next region. The output of that one goes to a different relay center in the thalamus, which projects to that region. The output of that one goes to a different relay center. So they're saying this might be the main feed-forward pathway. The main feed-forward pathway is that every cortical region gets its input from the thalamus, and it projects to another region through the thalamus. And although numerically these are smaller number of connections than these, they are much stronger. They're much more impactful. In fact, if you think that the cortex works on a single algorithm, the primary sensory regions always get their input from the thalamus. They don't get anything else. They don't have an equivalent to this input. They just get this. And so a region must be understood how it gets input from the thalamus. And they're saying every region has to be understood that way. That is the primary input to every one of these regions. It's through the thalamus. And this is the secondary feed-forward path.
What does it mean to be like primary, secondary, one to be stronger or weaker?
The arguments are that this seems to have more of an impact on the performance. You know, if I'm going to look at the effect of activity on this pathway versus the effect of activity on this pathway, this is what they call a driver. These synapses form big synapses near the cell body. They make the cells fire. They're more of a, this is just like from the eye, when you go through this part of the thalamus called LGN, which the eye projects to these cells here, which are projected here. These cells drive all of activity in V1. That's it. That's where they get their activity from. It's from the thalamic relay cells. And no one questions that for V1, for the primary sensory region. It's very clear that these cells drive these cells. Nothing else. Here they're saying, it looks like these cells drive these cells, and these cells drive these cells, and these cells drive these cells. So it's like a very powerful driver. And this is copacetic with the whole idea of a common cortical algorithm. And we can say, like, look, a region of the cortex doesn't know what it's looking at. It doesn't. These all look the same, and they all work on the same principle. So if one of them is going to be driven by thalamic input, the other ones are going to be driven by thalamic input. And this is how it's connected. These are often called relay cells because it's almost not quite, but it's almost like, one fiber comes in here, it makes this one cell fire, and then get another fiber coming out here, and it's like it relays the signal. But we know that's not true because it's very complex, but it looks like that. And Subutai did a paper, was that published?
It was in cosine.
A cosine paper. We've spent a lot of time speculating what's going on here. What are these cells doing? What are these relay cells doing? And we can talk about that. So this is an alternate view. It's not like both of these exist. This feed-forward pathway exists and this feed-forward pathway exists. Sherman and Guillory have been arguing this is the main feed-forward pathway. I think that's almost certainly correct, although most neuroscientists just want to be pissy about it and argue against it.
Because that's in all the textbooks.
This is in all the textbooks. They don't want to rewrite the textbook.
But there's some really interesting data now that if you cut off these connections, I think Murray Sherman showed this. If you cut off this one, it has a huge impact on the activity here. Whereas if you cut off these connections, it has a pretty minor impact on that.
So they call these drivers and they call these modulators or something along those lines. Yeah.
From lesion studies, have they determined what the relative role of each of those little subregions in the thalamus are, other than projecting to that area?
They don't look like they're doing processing. It's not like they will function. That's why they call them relay cells. There's not enough neural machinery here to look like it's doing a lot. Like when you get up here, you got tons of cells all doing all this complex stuff. Down here, it's just like, oh, these little relay cells and some inhibitory neurons. And we know it's not true. We know that it's doing more. But if you just look at the amount of neural machinery here, it's tiny compared to the amount of neural machinery here. And so, but it seems to be critical, and I'll come back to what it might be doing in a moment. But as a general rule, there hasn't been a lot of like focus on, well, I should say, general rule, like, oh, the function of these things is X. It's more like they look at them, they see what the response properties are, they do a lot of analysis of it, but it's not like someone said, oh, this is recognizing, faces. It's not like that. It's more.
So they can't correlate to any particular loss of visual behaviors.
Well,
there are some, attention would be.
These are associated with attention. I'm going to come back. I'm going to get more to it in a second here.
Okay, sorry.
Right? No problem. By the way, if you lose these connections, you're dead. I mean, you're a vegetable. The thalamus, although it's small, it's like the size of a small bird's egg. There's two of them on each side of the brain. It's relatively quite small compared to the neocortex. They're small structures. But they're so critical to the processing information flow in the brain that if you remember that woman in Florida who was brain dead and there was the big debate about letting her off life support, Terry Schiavo, her thalamus was destroyed. And without a thalamus, you're a vegetable. There's nothing going on in your head. That's where all the information's going.
Do all animals have a thalamus? Or just mammals?
Oh, no. Well, the neocortex is a mammal structure.
Right.
Birds have thalamus.
Yes.
But is thalamus part of the neocortex? No.
Good question.
It's all these things have precedent. Birds have structures which are somewhat similar to the neocortex too, or I think.
I would say you can't understand the neocortex without thinking about the thalamus.
Yes, that's the way we put it. And the way I've come to think about it is the following. And Murray Sherman agrees with this. He likes my analysis. I say, look, the way to think about this is that these cells here are really another layer of cells in this region.
I think it's part of the common cortical algorithm.
Yes.
Is these predictions.
Yeah. Then the question is, why are they located here and not up here? And it's clear that the brain wants to put these all very close together. Or at least it wants to have these relay cells all close together. So up here, remember, V1 could be a big area, right? And there are no cells that project all the way across V1. That doesn't occur. So V1 is a bunch of separate little things that are not really connected together. But here, these things are all very close, and so it gives the brain the ability to do something together to all the inputs to this at once. And so it's basically bringing the lower layer of all these things down to the central area here where it can do something on them, and we have some ideas what that is. But it's really to think about, even though birds may have had a thalamus, everything exists in some form in a previous animal. But the way to think about this in a mammal, and all mammal cortex looks like this. Every animal has this structure, basically. Is that this is really a part of the cortical algorithm, and it's really just another layer of cells, and it's now located centrally here because it was a point of being central control. So
does it control the output as well? I mean, does motor output goes to
let's talk about that in a minute. Okay. So what does this look like in the layers of the cortex? Okay, remember over here we had input to layer 4, projects to layer 3, layer 3 projects into layer 4 into layer 3. Here it's different. Here's a thalamic relay. This is part of the thalamus here. Okay, this is one of these little guys here. And so I'm going to show like this one right here. That's what I'm showing here. Okay. And so this cell, where is that? That cell is in layer 5. Okay, so it's not layer 3, it's layer 5. So layer 5, in this case, is the feed-forward output of this region, where layer 3 was the feed-forward output over here. This is layer 5. And it projects to this relay cell, and then projects back up to layer 4, primarily. it turns out that all these things make other connections too, but primarily layer 4. And so layer 4, again, projects to layer 3, and layer 3, in addition to going forward, layer 3 cells become the to layer 5. So if you follow the activity, it goes from thalamus to layer 4 to layer 3 to layer 5 to thalamus.
And then back to another region?
Then to the next, and just repeat that again to the next region, the next one.
Oh, wow.
So there are 2 feed-forward pathways, one that exits from layer 3, and one that exits from layer 5. Layer 5 goes through the thalamus. Layer 3, I can just draw it right here. Layer 3 cells would look like this, right? Also project to layer 4, but directly. That's the one I showed over there. And that's repeated again and again. So 2 forward pathways, one through the thalamus, one direct. One seems to be the driver, more important. One seems to be more of a modulator, less important. The textbooks mostly talk about this one, but this seems to be the most important one. And now to make things really confusing, these same layer 5 cells that project to the thalamus, which then project to the next region, that same axon, which is coming off this cell, splits into 2 parts. One goes to here, and one goes someplace else in the brain. And the general rule is the other place it goes is to something related to motor behavior. So in the part of the cortex that represents your, that's most connected to your body, which is this band right across here, that is what they call the motor cortex. There are these cells in layer 5 that are really big, and they're called BET cells. They're layer 5 cells, and they're just big. The reason they're big is because they have to project all the way down to your spinal cord. So when people talk about motor cortex, they said, oh, this is motor cortex right here, and it's got these big cells in layer 5, and they project to the spinal cord. That's it. That's the motor output of the brain. But now they discovered that that's not really true, in terms that every region seems to have layer 5 cells. They're just like BET cells, they're just smaller, and they do the same thing. So in visual cortex, back here, the cells in layer 5, this is like, even if this is V1, primary visual cortex, it has a motor output. And V2 has a motor output. The cells in layer 5 project to, in this case, they project to the part of the brain that controls eye movement. So even every region seems to be doing input and motor output, input and motor output, input and motor output. As far as I know, there's no exceptions to that. So it's not like the information goes chunk, 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 and now I have an output. It's more like every region is doing sensory input, motor output, sensory input, motor output. It's a sensory motor system everywhere. And this is the way it looks. Now this leads to a very confusing question. How could the output of a region be both a motor command and a feed-forward command to the next region?
And are they outputting to the same region? Every region has an output that goes to the same region? Or like different regions?
This is, you talking about this output or this output?
That output.
The left one.
The left one?
Yeah.
No, they all go different places.
Different places, always different places.
Yeah, because like the visual one projects to the things that move your eyes, right? The auditory, if this is auditory cortex.
But I mean, like all those first 3 regions, like the 1, 2, 3, they're all vision.
Okay, I don't think we know, actually, I don't know the answer to that question. I don't know the answer to this question. Is there a differentiation between where the layer 5 cells here and the layer 5 cells here and the layer 5 cells here project?
Yeah, they are all like vision regions.
You know, it's complex. I would probably say that you're going to find weird stuff going on. For example, you know, they might also, they project the superior colliculus, but superior colliculus is also involved in head movements. So, you know, and so the auditory regions project, so like if I'm listening, I hear something, I might involuntarily just turn my head like this to hear it. You're not even thinking about it? That's because the auditory cortex is saying, I want to move the ears to hear better, to change the input. And the visual cortex may be doing the same thing, you know, like this. So I think in general they say, there's some simple examples where they say, oh, this region projects to the body, The visual region projects to the superior colliculus, which is vision. The auditory region projects to various things also related to movement related to auditory. But I bet you I don't really know beyond that, and many of these, I think many of the regions in the cortex are uncharacterized. They don't know. It's just really confusing. There's also parts of the brain in the center, the old parts of the brain, the basal ganglia, which also control a lot of different movements. You know, like imagine chewing, right? Normally, you don't have to use your cortex to chew. It's the old brain does it. You don't have to think about it, right? You don't bite your tongue, you just put the food in your mouth and somehow it happens. But the cortex can control it too. So, you know, the cat runs out and say, oh, I'm going to think about chewing right now. So that's true for almost everything on your body. And so who knows where these project? I think it's I don't know the answer to that question.
I have a slightly different angle on that same question. So V1 and V2 are both projecting out through the thalamus to control some motor command.
Not through the thalamus. They skip this branch, skips the thalamus.
Okay, skips the thalamus.
But they project through the thalamus to provide input to the next region.
If V1 and V2, say V1 wants you to look left and V2 wants you to look right, is there a mechanism for like deciding who wins?
I have no idea how that. Well, I'm not even sure that they would disagree. I don't know.
Pooling would deal with it.
I don't even. I think it's a question. I wouldn't even know how to answer that question. Is there a disagreement? I don't know.
Well, our voting mechanisms might reconcile some of that disagreement.
Yeah. I don't think the cortex, one side of the cortex will say go left and the other side will say right. Except in split brain cases. There are people where they've cut the corpus callosum and the other callosal fibers that connect the two halves of the brain. And those people are pretty normal, but it's like they have two brains. And the left brain and the right brain can make different decisions. And then only the left brain can speak. So the person sometimes will say, like, I don't know why I reached over there. I thought I was going to reach over here, you know. If the right brain could speak, it said, well, I reached over there for this, but the right brain can't speak. So, but in general, I think it's your sympathetic probably right. So our voting mechanisms really. The voting mechanisms we propose in the framework paper and the Commons paper, I guess the original Commons paper, essentially tries to get all these regions to agree on what it is they're doing and what it is they're seeing, the voting going on. So there is a feedback, there is another feedback pathway here that's very important that we can talk about in this framework here, this view here. And there are cells in layer 6 which project back to the thalamus. This is not a complete feedback. These cells do not go to the thalamus and back to the region below it. It's not a downward chain. It's just back to the thalamus. And so the same region that's projecting, the same relay cells here that are projecting to this region get a feedback from this region back to them. So this is the way for a particular region, like this region, can control its input somehow. It's saying, I'm able to send the signal back to the thalamus to tell the thalamus to change something that I'm about to get. But it doesn't go from here to here to here to here to here. It doesn't do that. It's just like some sort of control on the thalamus. There are a lot of these kind of cells. There are 10 times as many cells projecting back to the thalamus as there are from the thalamus projecting to this region. So the one that's projecting from the thalamus, this feed-forward pathway here, remember these are drivers. This is like a very powerful pathway. There's 10 times as many neurons projecting back, and it's not a driver. It's a modulator. It somehow changes the input here, but it's not like driving the input. It doesn't directly make these cells fire. It changes which ones are going to fire and how they fire. But there's a 10-to-1 ratio between these and the feed-forward pathway. So this is like these two classic feed-forward feedback. Maybe this is where the convolutional neural network ideas came from. That's the supertypization. This is more of the reality of what's going on, and that this sort of sensory motor, every system, this tells us every system is a sensory motor inference system. Every column, every region in every column is a sensory motor inference system. It's not just a feed-forward spatial, pooler, temporal pooler. And very complex functions going on in here, and it's creating behaviors everywhere. And so now we can see this is like a complete sensory motor system, this is another complete sensory motor system, and now we have to explain what information is one sensory motor system passing to the next sensory motor system, and why is it controlling the thalamus in doing so. Here's a bit more detail of what's going on in the cortical region. I've added a few more of the connections within this here. So now you notice they often demark differences between 5A and 5B and 6A and 6B. There are actually maybe 5 different cell types in 6, so 6A and 6B are only 2 of them. But you know, every scientist can come along and say, oh, I found 1% of the cells that do X, that's a new type. Oh, okay. I've highlighted a couple of new major connections which I haven't mentioned yet so far. I've talked about how the input comes into layer 4, the layer 4 projects to layer 3 projects to layer 5, I show that here. But about 40 to 50% of the synapses on layer 4 cells come from layer 6, that's this black line. 40% of the inputs to these layer 4 cells is coming from layer 6. These don't appear to be the main drivers, but that's a lot of synapses. And it turns out that layer 4 cells also project back to the layer 6 cells. It's a little odd, it's mostly the apical dendrites of layer 6 cells are up here, but the projection is the same. So you have this very tight coupling between 6A and 4. There's a similar tight coupling between 3 and 5. So I've already talked about 3 drives 5, but there's also a coupling that goes back this way too. And the basic theory that we're working on is this kind of coupling is the kind of coupling we would expect to see between grid cells and place cells. You can think of layer 4 as a little bit like place cells or sensory-driven representations of location or information. And so, but in order to get me to do the sensory motor sequences so that I can pool in layer 3, I would need to combine it with a grid cell location. So this kind of mechanism right here would allow layer 4 layer 4 could learn high order sequences, but it could also learn sensory motor sequences based on if those were grid cell like cells in layer 6. I say this because it's more complicated than this, but that's the basic idea. And so we focused a lot on a couple of papers which sort of highlighted these massive connections between these 2 layers because it could be key to understanding everything that's going on. So now we have the ability to say, okay, well layer 4 in theory could be learning high order sequences, it could be learning sensory motor sequences, layer 3 could still be doing pooling, temporal pooling over them. It's more complicated than that. And now I've shown that here's the feedback to a thalamic region from layer 6, and here's the feed forward from a layer 5 cell to the next thalamic region and the next region in the cortex and motor. So it's a bit more complete picture than I had before. So I think I'm going to throw out this one idea that we've talked a lot about the thalamus and how it's implicated in attention. That's a very fuzzy topic, and what does it mean exactly? And there's been a hypothesis about what it does. I've introduced a new hypothesis which I really like, which sits on top of that. It's copacetic with attention, is that there is between the thalamus and the cortex, when you attend to things, there's an established frequency, a gamma frequency. Meaning these 2 cell populations cycle at a rapid rate together, like 8 hertz or whatever it is. I think gamma is 8 hertz. And so clearly when you attend to something, when these things are working together, they're synchronized somehow. And one of the hypotheses I have, one of the things we know about grid cells. So we hypothesize that there's grid cells in the neocortex. We're hypothesizing some of them are going to be down here in layer 6. Grid cell equivalents. They're technically not grid cells. If you define grid cells as being in the mental cortex, they're not grid cells, but grid cell equivalents. the question is, how do grid cells work? And one of the hypotheses of how grid cells work, how do they integrate over time, how do they integrate motor behavior to update a location? So I'm not going to talk about grid cells in detail today. There's two basic theories about how grid cells might work. One is based on an oscillation frequency. And how there's this sort of interference between frequencies of these cells that allow you to, when you move, how does the grid cells know to update their location? Well, when movement is associated with a change in frequency, and when you change the frequencies, the active grid cells move and change.
Are these cortical grid cells or these?
These are cortical grid cells. We're all talking about neocortex here.
Okay.
No entorhinal cortex.
Okay.
So this one theory, and I really, really like this theory. That's why I keep saying, because that's why I'm going to get it in here. Is this oscillation between the thalamus and the region could be a way of changing scale and of time and space. So by that I mean when you attend to something, you might think about it as like, oh, I'm looking at Subutai, he's either close to me or farther away from me. But how I would, if I think of him as an object that I can move my eyes over, a sensory motor object, and recognize him, the angular movements of my eyes will be different if he's further away than he's close. I don't want to have to learn that different ways. And one of the things that could be happening here is that essentially the thalamus could be, the cortex could be controlling essentially the scale of movement, like a movement command, how much it actually moves, could be controlled by the thalamus, this oscillation. So I've already speculated this back in my book 14 years ago about how the thalamus, some of these cells in the thalamus could be changing scale of time, so that like when you recognize a melody, you can recognize it faster or slower, or I can play back a sequence that I've memorized faster or slower. So I have to have a way of changing scale of time in the cortex. And these cells look like they'd be perfectly positioned to do that. And changing scale of time is really the same as changing scale of space. They're really kind of the same thing. And so the idea that the thalamus could be this role that's not only you attend to certain things, but attention means you're scaling space and time to adjust to the new location of this object, or the new tempo of this object, or the new position of this object. It's a little more complicated. It's a complex thing to absorb, but the idea that this is really a scaling function in addition to an attentional function. I think there's a lot of merit to that, because this scaling property has to occur, that you have to be able to scale in time and space. So all this is complicated, but this is what the real brain looks like. So I'm done here, but yeah, go ahead.
Do you think that scaling is a way of attention? For example, you just give a lot more importance to some things than to other things, or do you think of scaling and attention as two separate things?
I think they're tied together. There are multiple components of what attention means.
When he says scaling, he's not talking about adding a multiplier, like scaling a value. That's not what he's talking about. Physically scaling things.
But could that be also? Do you think that, for example, you can scale things? You're thinking about scaling to the right proportion, but you could also scale to a greater proportion. And I don't know. Can have like when we have to think about latent representations, which are much bigger for one thing than they are for another thing.
Yeah, let me give you an example. So we all have a smartphone in our pocket here someplace, right? And there was a company that came in here recently. Actually, they now own the name Palm, and unrelated, totally unrelated. And they're making a teeny little smartphone, teeny. They're trying to sell this, and it's like you look at it, it's like a teeny version of this, and it does everything. It's just like a version that's smaller. And well, I have learned how to use this phone. I know how to type on it, use my thumbs, and I know how to scroll things up and down. I've learned how to interact with this phone. You give me this smaller phone, I'm able to interact with it right away. I can type on it. My thumbs aren't making the wrong movements. How does that do that? I've learned certain movements here to type, and now you give me a smaller keyboard and I can type. How did that occur? How do I know only to move my finger a little bit? and not as much as I would normally move it. So this kind of stuff is happening everywhere in the world we deal with. We're constantly scaling things both in space and time. Things are faster, slower, and larger and smaller. So movements have to be scaled. I'm able to operate that thing without relearning. So, and even though I've never done it before. So clearly that tells me I have learned how to operate my thumbs on a keyboard on a smartphone. At the same time, I'm able to turn a knob which makes my thumbs move further or less. It's like I can type on an iPad and my thumbs move fast, a lot further. So this kind of stuff you can convince yourself is occurring everywhere in the brain. So it's an inherent property that must be addressed, system-wide. So that's where the beauty of having this in some central location. I can scale the entire input to this region in one spot, even though I can't do it up here because there are no long-range connections that go from this side to this side. But I can do it down here.
So when it goes through several different regions, you scale once and then you send that information.
No, actually, it doesn't appear to be that way. There are cells in the thalamus called matrix cells. I didn't talk about them. They are not the relay cells. Okay. And the matrix cells also project to the cortex. But when they project to the cortex, they project very broadly over a single sensory modality. This is what got me a long time ago. I mean, so for example, if I look at the matrix cells in the visual regions, they project across all of these guys. If I look at the matrix cells in the auditory region, they project across all the auditory regions, everywhere, up and down the hierarchy, left and right. So it's as if you're applying and I argued that was time scale. I was arguing that the matrix cells are providing a timing signal to the cortex. I still believe that's true. And so they're providing a timing signal. The way the cortex, and the matrix cells, just to confuse you even more, these matrix cells, what they do is they project up into layer 1 very broadly. So what they're doing is they're providing this timing signal that the different cells in the different layers can use to regulate time. And if I were going to adjust the time, I would adjust it for all of them. This is consistent with the 1,000 brain theory hypothesis. It's not like this guy's running a one-time scale and this guy's another time scale. They're all doing the same thing. They're all running at the same time scale. He said maybe in the vision, this one's looking at details in one part of the visual space. This is looking at more broader parts of the visual space, and so on. Yet there's still this sort of hierarchy too. So we have to explain all of that. But anyway, the idea that there's these that you would be scaling time or scaling attentional time and space would be applied to all the different regions in that modality. But I don't see matrix cells jumping from the visual region to the auditory region. So all the auditory system is going to run on one sort of time frame, and all the visual stuff is going to run on another sort of time frame. You can't really visually attend to two different things, and you can't really auditorily attend to two different things. You jump back and forth. And there's a lot of evidence to support this. When you change attention, these cells change, the activity changes, the frequency changes. So these are all hypotheses that I don't exist, I think, anywhere else, but I'm pretty confident that they're basically right. So you can ask more questions, but the bottom line of all this is, I think the most important thing for our work right now is that when we think about the ultimate AI, it's not going to be 100 layers of sort of pooling or max pooling or something like that. It's going to be, complex sensory motor functions of which you might have multiple levels, a few levels in the hierarchy. But every one, the 1,000 brains theory says that every one of these things is doing a complete system. So you could build a complete AI system that just has one region. It may be limited, but you could do that. And where you can't do that with convolutional neural network. So this is a much more powerful system. And today we're not doing any of that. Our work today is right now, the super time you guys will work on is just trying to introduce the property of sparsity. But I believe that in the future we're going to have to introduce all these concepts to do it through AI. I don't think these are optional. You don't have to be done this way. You can do them other ways, but these functions have to be there. Scaling the time, motor, sensory motor inference, all this stuff has got to be there. Okay, I'm done, unless there's more questions. Yeah?
You said there are a couple of theories about what the thalamus is doing.
Oh gosh, none of them very good.
No?
How would you say
Well,
something?
about what the thalamus is doing?
Yeah, what are the other theories?
Well, one other one, which is, if you think about what attention is, it's sort of focusing activity maybe in one part of the input space versus others. a more general version of this is to think of sort of a complex routing mechanism or transformation mechanism where you have inputs coming in and you pay more attention to some subset of it that doesn't have to be physically contiguous.
And we've led in that area too. I mean, this idea that the thalamus may be a route changing thing has been around for a while, but
yeah, so if you think about temporal scaling, but you could also do physical shifts and stuff like that. So there could be all these transformations that occur.
Subutai and I, and what's her name?
Carmen.
Carmen was at MIT, now she's Florida, I think. There's a lot that's known about the really detailed architecture in here, and it's very unusual. And so we have postulated, we've looked at that detailed architecture and postulated how this rerouting stuff could work. So the idea that, literally when I attend to something, I really just want to turn off the inputs from elsewhere. Like I don't really want to see everything in the background here, I just want to see this thing. And so there's this physical routing attention in addition to the sort of scaling properties appropriate for that. Most people who, if you read what they write about the thalamus, they'll say things like, oh, it's a driver, it's a modulator, it's modifying the inputs. My theory is that these inputs will modify these inputs. It's like, yeah, okay. There's a lot of like fuzzy hand-warming stuff, hand-waving stuff. But the kind of detailed theories we're talking about here are very, very unusual.
Are you familiar with like a multiplexor? Like
in computer hardware.
In computer hardware, yeah, like a multiplexer. You could send in a pattern, and based on that pattern, it changes the routing of inputs coming in. And different patterns will lead to different multiplexing.
So like, they talk about,
that's what we think is.
They talk about this relay cell here, right? You got this input goes to this relay cell, and this relay cell projects to here. Well, what Subutai is talking about is that's really a fib. It turns out that if I look at multiple relay cells here, an input actually connects to multiple ones. But it only appears if one of them is actually doing anything. So they say, well, that's the relay cell. I don't know why these are connected to that. And what we think is going on, we're saying, well, under the correct conditions, this can be routed, and that one can be turned off, and this one can be turned on. So you really can multiplex, you can just reroute these signals. It's still one-to-one, but the routing, there's no like convergence on these cells. It's not like these cells are recognizing a pattern, but it's literally like you can route the signal through different ones. And there seems to be a lot of evidence for that. I mean, you and Carmen wrote that paper about that.
Is it the cosine paper we're talking about?
It's cosine. It's not a full paper.
It's a poster, right?
It's a poster, and we're slowly working on a paper for it.
Yeah. I want to publish this idea, even if I'm looking for an opportunity, even just have like some sort of a note, like here's an idea what the thalamus might be doing, because I would like to write up these time scale and space scale hypotheses. I think that's correct, and I'd like to at least get it out there in the literature world sometime. But I don't think I'm going to be able to write a full paper on it. So it would be more like if there's an opportunity saying, here's a research note, letter to the editor or something, like here's what might be going on there without like doing experiments and proving it and stuff like that.
So you're saying the layer 6 feedback cells could also impact the oscillations?
I think they did, I think the oscillations would determine the scaling.
Yeah.
And what creates the change in oscillations, I think we'd have to go look at the literature on that again, but clearly it's going to be some interaction between the cortex and that region, and as far as I know, the layer 6 cells are the only one that make that interaction.
Yeah, I think the mechanism and the anatomy of the thalamus could support something like that.
Oh, I think definitely support it. Every time I read about it, I was thinking about this. And so that whole reticular formation and
exactly.
the way that
they have gap junctions.
Yeah, the gap junctions are all set up to do cycles.
Yeah.
And no, I think it all fits. I think
if the oscillations in the relay cells change, it's going to change oscillations in the.
I'm not even sure the oscillations in the relay cell change. All we know that there's oscillations that are occurring when you look at area potentiation. I'm not sure they've measured them in the actual relay cell. I don't know if that's true. But if you look at the overall activity in this area here, you stick in a probe and say, what's going on in this area? You'll see these waves, and you see these waves. So I'm not sure I don't know if we know, I don't know if they know that the relay cells actually oscillate. I don't think that's even, that's not important to my theory. My theory is that that somehow the oscillation, you want to establish an oscillation across all of these regions that will uniformly impact time and space scaling and motor scaling for every region in that modality. So it's more like how this thing projects back to here with that frequency, which would then impact how the grid cells, when you get a grid cells get a motor command, how much do you actually move? It's the question. And also about the timing of sequences. Anyway, I'd love, before I croak, I'd love to write that up sometime.
How fast do those oscillations change? Because it feels like
They change pretty quickly.
okay, I feel that they would have to because it seems like you constantly be like rescaling things.
Yeah. There's not a lot of literature on this, but we did read something about that. I can't remember. Maybe Marcus, you remember? I don't know. But I was testing that idea because they have to be able to switch. It's sort of at the same rate at which you can switch attention.
Yeah.
And my recollection was, don't ask me where I read it, is that they can change pretty rapidly. It's really, when you attend to something, it's as quick as you can attend to something, that frequency is established. Remember, a lot of times if you're not attending to something, there isn't a cohesive gamma frequency between them. It's when you attend to something that the cohesive gamma frequency kicks in, and that can happen as rapidly as you can attend to something.
Seems like something that would be very easy to like test experimentally, take and measure this oscillation. Instead you like focus on.
Yeah. My recommendation to you is never ever say to an experimentalist this is something you test.
Easy means 3 years.
Yeah, and also you don't really remember the thalamus. The thalamus is way inside the center of the brain.
Yeah, sure.
You can't just get to these things, and it all looks like bloody tissue. And how do you even know where you are? And how do you measure this stuff? It's really complex to do these things.
And you'd have to do this in a live animal while they're doing an attention-related task. And you train the animal to do the attention.
So
there's a whole bunch of
in the beginning, most of these experiments were done, almost all these experiments were done with slices, where they would take a brain, they would move the animal, they turn a slice, and now you've cut through the brain, and now you're looking at the profile just like this. And then you can probe here and you can probe here, but this is a slice, you've lost all these other connections, the brain is isolated from a body, it's not, so you can say like, okay, you wouldn't be able to do any of this, because as soon as I said the animal has to be awake and attending and doing some task, and you have to know the animal's doing that task. You can't ask a rat, hey, what are you thinking right now? Attend to this, attend to that. You can't do that. The rat's just going to do its thing. It's going to go, so then you have to deprive the rat of food, so it's super hungry. All it's going to do is think about one thing, and you hope it doesn't think about something else, and it's really hard.
Can you do it in humans with like electrostimulation? Can you deactivate some?
You cannot stick a wire into a human.
Well, that's what Elon wants to do.
Yeah, but he's only doing that now for patients who already have a medical need to have their brain exposed.
So I mean, like epilepsy patients.
Yeah.
So can you do this kind of experiment?
But they can only do it in the surface of the brain right now.
Yeah.
The thalamus is really deep in.
They do probes, for example.
You have to stick something from here all the way down into here.
Yeah.
And kill everything along the way.
And so there are experimental procedures where they do stick probes very deep in. If you're trying to cure tremors from not Alzheimer's, Parkinson's. Thank you.
Yeah, there was people in Cambridge and Oxford are doing that.
Yeah, I know a woman who just had that done. She's got surgery where they stick a probe way into her brain, down to the some part. Yeah, something like that. Yeah, that might be it. And then but they will never do anything like that, and that's a pretty radical surgery. Right. And they won't do that unless you need a radical surgery. Right. You can't just say, oh, by the way, your whole brain's open, let's go stick some probes into it. So it's really hard so now what you can hope is they can do this stuff with fMRI. And fMRI you can use on live patients. The problem with fMRI is two things. One is the temporal resolution is not what you want. Meaning you can't really pick up small time differences. It's fMRI picks up it's a blurred time pattern. And also, the size of the region you can measure is getting smaller. They're now down to sub-millimeter, but which is still a lot of neural tissue. You can't look at individual cells at fMRI, and you can't look at them on the order of milliseconds or something like that. You're blobby effect. So these experiments are really hard to do, and although it's okay for us to say this could be testable, we say this theory about the thalamus is definitely testable. It just may not be testable easily, and it may not be testable using today's techniques. But in theory, it's testable. It makes predictions that can be validated or falsified.
But when they take measurements of the brain, like I think I've heard of things like, oh, like something's happening at such a frequency. Like those frequencies that they measure the brain, what are they typically measuring? Is it not this thalamus thing?
Well, they can measure that. You can measure some of those frequencies with scalp recordings. You can put EEG scalp recordings on, and you can get these massive, like when I say massive, when you go through the scalp in the skull, you can measure the activity of the upper layers of the cortex in some regions over large areas. So they can see frequencies that are occurring. So like, during sleep, you'll see one set of frequencies and another. If you want to get something more refined than that, more specific, like okay, what's going on in this little area right here, then you got to stick a probe in, and then you can
the electrical activity.
Yeah, you can measure the potential, the local field potential they call it, which is like all the cells around this probe. I can sum up their electrical activity, and sometimes I can pick out individual spikes. So there's a slew of different techniques. They all vary in terms of their invasiveness, their difficulty, the preparation, what can be done in what animals, whether it's slice or vivo, the animals can be free moving or not free moving, because many of these experiments, even if they stick a probe in the brain, they have to lock the brain down. It's crazy. Now they have these experiments where rats are locked into position, their heads are fixed, but they're running on a ball, and the rat is in a virtual reality environment. So they're projecting around the rat as if the rat was moving freely. So in theory, the rat thinks it's moving freely through the world, but in reality, it's fixed. So they can put the microscope right on top of the cell, the brain, and look at it. These experiments are very, very hard. So mostly what we've done is we can use logic to say something has to occur, like pooling has to occur, these different types of pooling have to occur, these different types of sequences have to occur. These are not speculations. These are deduced properties of the brain. This kind of scaling I'm talking about has to occur. It's not an option. I prove that it has to occur. Then we can try to map out, like, well, where could these things be occurring? And what anatomy supports that? And so we can make a lot of progress without actually doing these experiments. We can just look at all the existing data and deduce what are the options, where could this be occurring? Yeah. By the way, I should mention something about temporal pooling, since that was one of the things we originally started with. This idea of this many-to-one mapping, temporal pooling is very memory intensive, so you can't map 1,000,000 patterns into one pattern. You can't do it. There's not enough synapses on the neurons to do that. So we did a lot of testing on this. The sparser this is, and the sparser these are, the more you can do. The threshold of the dendrites matter too, but there is a limit. And one of the things I thought about this is when you learn like melodies, and melodies can be quite long, and I asked you to name the melody. So you're hearing the melody, you recognize the melody, you say, oh yeah, I know that song. Then I ask you to name it. It's often very hard to name the melody, even though you recognize the melody. And often if you are able to name it, it's when the melody gets to some repetitive refrain or a chorus or something like that, or you hear the name. So what I think is going on there is, in general, the temporal pooling layer can't pool over all possible inputs. There's just too many. And so it's limited. It's much more limited than the sequence memory itself. The sequence memory, a little cortex can learn hundreds of thousands of elements, but this guy can't. So it's a limiting function in the cortex, and it's just something to bear in mind. And we did capacity testing on that. It's not small. It's not like trivial, but it's one of the limiting things. And as I get older and I have trouble remembering things, I don't have trouble, of course. Like all people have trouble remembering things. You know the thing. You know the word. You know the sequence. You know the melody. Whatever. You just can't name it. And so I think this system here, the temporal pooling system, is always working on the edge of its abilities. It's right on the edge of we're pushing it. So as things decay, you lose that first. You don't lose you know the word. You just can't think of it. You can picture the person, you can picture what they do, you can think of all the things that you know about that person, you just can't remember their name. It's like, that's crazy.
Well, if you think about what was necessary for survival, being able to recognize a sound that is a danger,
Yeah.
a pattern of a predator up in a tree, whether it's a danger or not, you're supposed to react before you actually could, well, that's a leopard.
Yeah, exactly. Yeah, or it doesn't matter if I name it, as long as I know what it is.
Or what to do about it.
And what to do about it, right? Yeah. So again, it's like a melody. I recognize all these melodies. I just can't put the name on them. I mean, that's a placeholder for recognizing people's faces or people's, who they are. Places.
So the ability to recognize a snapping twig versus, something flopping in the water, something like that, would be something that we want to earlier, faster reaction, and then everything built on that would be cognitive, right?
Yeah, yeah. Well, I'm not sure I'd make a hard distinction between cognitive, but you're right. I think we're saying the same thing. I think we're saying the same thing. All right, I'm done. I don't. By the way, unless someone wants an additional talk about some particular topic, I'm not planning one. But if you want an additional topic, just let me know what it is.
Okay.
I got one. Which I don't. But inhibitory cells.
We talked about that.
We talked a little bit about that, but in the thalamus and all the different kinds.
In the thalamus. We could do a very detailed talk about the thalamus. That's really now we're getting down dirty. I'd be happy to do that. I think it's getting further and further from what would be relevant for the work that's going on now. But we could do that.
Another thing that would be helpful is an overview of the thousand brains theory.
Oh, sure.
I've read the paper, the content which
the
was
paper
available.
didn't work as an overview?
No, but when you say it and when we discuss and then we can raise questions and all, it helps.
Sure. You want to review that paper?
That would be a good idea.
We could do a journal club.
Yeah, journal club.
Yeah, journal
club. I'd be happy to do that.
That would be good.
The thing that's often that's difficult, that's not covered in the paper, is going from SDRs and sequence memory and active dendrites and all of that stuff that we talked about, and the thousand brains theory, that's not really covered anywhere.
Yeah,
that's a great point.
Those things are not linked.
Well, but
we know how they're linked, but they're not directly linked.
Yeah, I mean, in some sense, I view our work, our opus of work here, as a series of papers. It starts with the neuron paper, and you have to sort of know that, and you need to know the SDR paper, and then you need to know the temporal memory, and you need to know the comms paper, and then you can learn the thousand brains. But you can't write all that up in one paper.
Right. Yeah.
It was hard enough to write those papers, even covering what we did, because one of the first rejections I got on the neuron paper, we got on the neuron paper, we were covering too many things. We're talking about too many stuff. You need to focus on one little thing, and the problem with our work is it's very hard to focus on one little thing. Right. Because it's the system that comes together. And this is a problem basically that neuroscience hasn't had an equivalent to theory like physics or cosmology has, where people in cosmology can come up with grand theories about time and space, and everyone says that's wonderful. We come up with grand theories about the brain. People say, oh, it's too much. You shouldn't do that. It's impossible. Don't even try. So, anyway, we can do a thousand brains theory. That'd be fun.
I don't know if it's a basis for a talk, but it does relate to trying to do function inform and look at the anatomy. But do we have a notion of how synesthesia takes place?
We don't, but I know what some other people have said, and I think it's very, very. This is a side topic. I can tell you right now. Mm-hmm. So I think there was Ramachandran who came up with this idea. I think it was. Remember here I said this is vision, 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 right? At some point that stops. And then you have auditory, or you have something else, right? Because there's other parts of the brain. There are parallel, connections that go for a millimeter, sometimes several millimeters horizontally here. And what Ramachandran has proposed for synesthesia, I'm not sure everyone knows what that is when you mix your senses. My father had it. I love this story because I only found out at the very end of his life. When I was growing up, my father didn't like beets. And I said, why don't you like beets? And he goes, oh, they have an oi taste. And that was like the running joke in our family, that the oi taste. Only when he was like 90 years old or 85 years old did I understand that beets sounded like oi. So when he ate beets, he heard oi. It wasn't like that wasn't his description of the taste, that's what he heard. So he had synesthesia. Anyway, what Ramachandran proposed is that normally you would not want any connections between these two. And so what he suggested was, perhaps what's going on in synesthesia, you do have these connections that are just crossing over between, let's say, visual and auditory. And he gave evidence for this. He showed that the classic synesthesia symptoms align with regions that are the two different types that are right next to each other. And so when you're getting activity here, it activates activity here incorrectly. And that's why all of a sudden something tastes or sounds. My father's taste system impacted his auditory system.
So that would explain like developmental synesthesia, but apparently it can also be drug induced too. So
I don't know about that. I don't know. I have no idea. I don't know.
Hallucinations.
The hallucinations may not be the same as synesthesia. In hallucinations, you're imagining things that aren't there. Synesthesia is real. These people, that's the sound of beats. It's not like he's hallucinating it.
Right.
That's what it is.
Right.
Just like we say beats are red. Well, why are they red? I don't know. I'm writing about this book, but point is, they also have a sound to my father, so that's it.
I've heard in kids, when kids are growing up, they have this synesthesia, this property of like mixing up the senses, and as they grow older,
I haven't heard this.
they kind of lose.
It would make a lot of sense.
Yeah, it does make sense. Yeah, I didn't hear that. I didn't know that.
So like kids always make this fun analogy. That's fun for us, but for them it's like.
And for the people who can see auras, there could be a synesthesia.
Could be. I don't know. Synesthesia tends to be very specific. Certain things have certain other things. Like the number 3 is yellow. It's not some fuzzy thing. It's just letter 3s are yellow. And the great thing about synesthetes is they think everyone else knows this. They don't think it's unusual. They go, you don't think that? You don't know that 3s are yellow? How could you not know that? It's like saying 3s are made by 2 loops. Like, you didn't know that? How could you not know that? It's so obvious. It's great.
For me, like it's obvious for me that left is connected to right, that's connected to odd numbers. And these are things that were always obvious to me. And
Is that right? Is that right? It's so obvious to me.
Yeah, it's obvious to me, but when I talk to
say that again.
Like for me, like left, it's connected like to red, and it's connected to odd numbers. And that's the way pair is connected to blue. It's connected to even numbers. I mean, for my head, this.
So that's type of synesthesia.
Yeah, and I always tell this to people, and people, no, it's not. It's not like that.
I don't have those connections.
Yeah,
I don't make those associations.
Well, the spectrum they usually display red on the left and it goes to blue to the right. So I'm just wondering.
Well, that's what Ramachandran pointed out. He pointed out that he could point out the specific locations of certain sensory representations were adjacent to the other ones that happen to be associated with it. So you could read his paper.
Which what's the name of it?
Ramachandran. I forget his first name. He's like this crazy. He's a bit out there. He's a neuroscientist. He has all these amazing. He's a neuroscientist and a psychiatrist. I don't know what it is, but he's
San Diego or something.
He's written all these books like about crazy psychiatric diseases and so on. He's just one of the guys who just he's a fountain of ideas. And
Is he like does like a lot about phantom limbs?
yeah,
exactly.
Yeah,
B.S. Ramachandran.
B.S. That's right. We don't know his first name. Two initials.
Is he still active?
UCSD San Diego.
I don't know. I think so.
Born 51.
I don't know. He's probably still active. Like 60s.
It's noon, so we should probably
stop. Okay.
Probably stop. So we'll just say thanks for people watching on the live stream, and hopefully you'll join us Monday for the Brains at Bay meetup 5.